Hi micro folks, this is Professor Carberry. We're doing a series of Introduction to the Microbiology Lab, so this will be Lab Intro Part 2. This is to go over the first day of lab, possible schedule of activities which may change depending on your lab instructor. The hope is by filming these videos, you can watch them before you come to lab so that when you come to lab we can just dive right in on the exercise. So first of all, welcome to Bio 440 General Microbiology. I'll be teaching some of the lab sections. Professor Christina Passerini will be teaching some of the other lab sections. But we try to coordinate our activities, and the grading will be similar, the assignments will be similar with just some minor variations. So it's really important if you are enrolled in the course that you arrive on time and don't miss lab, don't come late, because we have a full wait list. The only way we can add people is that if someone who's enrolled drops or doesn't show up. So if you don't show up for the first day of lab, we'll drop you so we can add somebody from the wait list. If you have an emergency, try to email me or call me as soon as possible to let me know so I won't drop you. If you miss more than one lab in the first 2 weeks, unless there's extraordinary circumstances, you will be dropped. And we will also be seeing folks that for accreditation in the entire semester, if you miss 4 labs or more, you will be dropped. And again, this is for accreditation purposes, which we can discuss later. So try to come early. Parking, traffic will be horrible, so try to leave really early so you can make sure that you'll get to lecture and lab on time. We'll start out with an introduction. I'll introduce myself, Karen Carberry Go. I'll be the lecture instructor, and then as I said, I'll be the instructor for some of the labs. As we mentioned, the only way we can add people is if somebody who is enrolled drops, and then we must add people in order from the computer-generated wait list. I'll take roll for those that are enrolled, and then I'll also take roll from the computer-generated wait list. But after this first lab, I'm going to ask folks on the wait list not to come to lab because of liability reasons. If you're not enrolled in lab and you get hurt, then our campus is liable. On that first day, I'll be passing out hard copies of the lab syllabus. It will also be posted on Canvas, so some of you will have a chance to read it ahead of time. And I'll be doing some screenshots going over the syllabus, and I'll be posting that on Canvas. The hope would be that within that first week you will have gone through the syllabus, and if you have any questions, let Professor Passerini or myself know so that we can get them cleared up. One thing on the lab syllabus, of course, is lab materials. So let me just show you a couple. This is the lab manual, Microbiology, Biology 440. It's very expensive, and just as a heads up, folks, we won't be using this Blue Door lab manual starting fall of 2020. It's just too expensive. For those of you that can't afford to get a hard copy of this lab manual, I'll have copies here in lab. What students have done in the past is they've used their cell phones just to take photographs of the pages. Also, there would be a copy on the second floor of the library, and they have Xerox machines there. If any of you have access to the older 2012 Bio 440 lab manual, if maybe a friend took the class previously, that lab manual works really, really well. Okay. Also, folks, do make sure that you bring a 3-ring binder because you're going to be getting so many handouts. That's one complaint some of the students have is I give too many handouts, but you'll need a binder. We have a 3-hole punch here in the lab. You want to keep those handouts organized and maybe with tabs so you know where to find specific handouts. The textbook that we're using is the free online textbook, OpenStax Microbiology. But if you're like me, I love having hard copy. I was able to order this hard copy of microbiology from OpenStax for $25. One of your colleagues shared with me that our SCC bookstore also has them available. Just check, folks, it might be cheaper to order them directly through OpenStax if you know money's a challenge for all of us. So trying to find the least expensive way to get your books. And do remember, folks, the entire book is free online. You don't have to buy a hard copy. For lab, the microbiology lab, it's truly a hazardous lab. We have the lab intro part 1 talks about all the hazards we have here. So absolutely essential safety gear that you have, and we call this our PPE, our personal protective equipment. When I say bring full PPE to lab, what I mean is a lab coat. And again, folks, the more surface area that's covered, the better. So if you can get long sleeves, right, and as long as possible, I mean, ideally down to your knees, the more surface area that's covered, the better. So you need a lab coat. You'll need closed-toed, closed-heeled shoes. These can be your street shoes, but just remember, if you like, when it finally gets warm, if you like to wear sandals or clogs, those aren't lab shoes. And what you can do is bring an old pair of tennis shoes in a plastic bag and store them here in lab as your lab shoes if you like to wear clogs or sandals. So lab coat, your closed-toed, closed-heeled shoes. Essential is indirect vented safety goggles. Make sure they're indirect vented. And you'll also need lots and lots of non-latex gloves. So they can be nitrile, they can be any other, I think there's vinyl, just as long as they aren't made of latex, because a lot of folks have latex allergies, right? So you'll need your full PPE. I would try to bring it week 2 or week 3, and then that way you'll be prepared when we absolutely have to have it. Furthermore, for a lab, we'd like you to buy your own microscope slides. These are available on the internet, very, very reasonably priced. One of your colleagues, I asked last semester at the end of the semester how many slides they thought they needed, and they had bought a box of 72 glass microscope slides on the internet. And they said this box of 72 slides was enough for their entire team of 4 people, right? So if one person buys a box and then everybody splits the cost in a team of 4, this is very reasonable. We have enough glass microscope slides. You don't have to buy glass microscope slides. I would suggest that you or a partner, you'll have a lab partner, buys a roll of adhesive tape, masking tape. We use this for labels. We do have tape in the lab, but it's only in one location, and so it's going to make you more efficient if you have your own tape for making labels and also for sealing your agar plates. We're going to be doing lots and lots of labeling, so I would really recommend you get a fine tip permanent marker. That's going to make your labeling so much easier. We're going to be doing lots of observations. You're going to be drawing lots of pictures. So, and we will want them in color. So if you can get a set of colored pencils, we do have colored pencils in lab, but of course the color you want often is missing. Or gosh, you guys, just go get a nice dollar box of crayons from the dollar store, right? And this will help a lot with your lab observations. Okay, let's see here. What else do we have? So paperwork is really important, folks. So the first day of lab, those of you enrolled will give you the lab syllabus, which includes a lab agreement form, which you'll be signing and turning in. There'll be safety rules, which you'll be reading, and initially you'll be turning those in. The most important piece of paper is the signed, completed lab waiver form, including emergency contact information. So we'll be collecting those. All of the paperwork will be collecting the second lab. And if that paperwork is not on file, you can't participate in the lab. So it's really important you get the completed signed paperwork in. For a few of you, probably not many, we'll also need to see your transcript verifying that you've had college level chemistry with a grade of C or better. It need not be official, but the transcript has to have your name, and we appreciate it if you would highlight the college level chemistry course. It has to be college level chemistry course with a grade of C or better. And in the lab and lecture syllabus, there's a list of pre-approved chem sections. If you took your chemistry course within Los Rios, I'll get a list of those of you. You don't have to bring me a transcript. We already have verification that you meet the prerequisites. All right. So then what we will next do is a group of 4 people is we're going to have you do a practice safety quiz. And again, this is just it's a little bit more efficient way of hitting some of the main safety issues. I'll be assigning you any of the safety videos that we have posted on Canvas. And then Professor Passerini came up with a more active way to explore the lab, get to know locations, also to reinforce some of the key safety features. She's developed a lab treasure hunt. So we'll probably break into pairs and take maybe 15 minutes or so for you to go and explore the lab. There'll be specific stations with questions for you to answer. And that way it gets you up and moving and gets you exploring the lab. So when we are referring to the kill area or the autoclave bags or the eye wash or the incubators, you'll have a better idea of what we're talking about. And then if we have time, what I'd like to do to close that first lab is to set you all free to do some environmental sampling. We're going to do some real simple sampling to prove to ourselves that microbes are everywhere. So we'll have cotton swabs, so if you want to swab surfaces, if you want to go outside and grab some plant material from outside or dirt, and what we'll do is we'll transfer any microbes from those substances onto the surface of an agar plate. And we'll be discussing these agar plates. It's like solid microbial jello, meaning the microbes will use the nutrients in the agar to grow and divide. We'll incubate the plates. We'll talk about how to make labels, how to process those plates, how to tape the lids to the bottom and invert them, and we'll stick them in our incubators. And hopefully within by the next week, you'll start to see some beautiful microbial colonies growing on your plate. And we can count the number of colonies to give us an idea of the number of microbes that grew from your sample. We can look at all the variety of colonies to get an idea of the variety of microbes that grew from your sample. So it's pretty amazing. Yeah. All right. So, and then folks, just very, very quickly, I just wanted to briefly describe what we mean by our Bio 440 course has the flipped lab format. And the flipped lab format, if I can say it, is what we do is we provide you with materials, with movies, with PowerPoints that you are to view before you come into lab. You're building your foundation, your understanding of what we're going to be covering in the lab before you come into lab. This way you don't have to sit for an hour and have us lecture to you and then you only have an hour to do the experiment. This way, by you doing that homework ahead of time, when we come into lab, we can make a very short intro, discuss safety issues, and then boom, you can get right to the hands-on part of the lab, the best part of the lab. So for this flip lab format, what I hope is that for each lab, I'll have a short video or movie, some kind of introduction that you are to view before you come to lab. In addition, we'll either have homework pages for you to fill in to verify, right, that you did your homework, or perhaps there'll be a little pre-lab quiz so that, again, when you come into lab, you're ready, you have a basic understanding of the new vocabulary, of the new concepts, so that you can dive right into the hands-on part. During lab, in the carvery labs, I will be checking to see that you attempted your homework, right, and there's points associated with that. During lab, we'll have you making and recording observations, and so we will be checking that you're recording your observations, doing your drawings, recording data, and then furthermore, to prepare for our lab exams, what you'd want to do is finish all the study guide questions that go with each of the labs. When you come into lab, we'll be giving you packets. I call these packets the lab study guide homework packets. They also include pages for your observations. On the day of our lab exams, you'll be turning in those completed lab study guide homework packets. You'll be graded on those. And the purpose of those lab study guide packets is to provide you a way to prepare for the lab exam so you can do really well in the lab exam, the lab practicals. Okay, so when people say, how do I prepare for the lab exam? I'm going to tell you, do the lab homework, do the lab study guide questions, and that should be a good foundation. Let's see here. It's really important, folks, that you try to stick up with lecture and lab materials. One way to approach Bio 440 is to think you're taking two different micro courses. You're taking a lecture course and a lab course. So unlike A&P, where the labs support lecture, often in microbiology, the labs are covering topics that we don't have enough time to cover in the lecture section. So it's an intense course. It is not linear like anatomy and physiology. So if you've had anatomy and physiology, you have a huge advantage. You know how to study. But those of you that have had A&P at Sac City, you might initially be uncomfortable in microbiology because, again, it's not a linear field. We tackle it more as a field in ecology. We're looking at all kinds of different topics. We're focused not only on the microbes, but we're focused on the environment. We're trying to understand how the microbes interact with their environment. And very importantly, we're trying to understand how do the microbes interact with humans, right? So it's a very holistic, kind of non-linear field. But I think if you hang in there and try not to get too discouraged or frustrated, I think a lot of folks get frustrated. Hang in there. Hopefully by the end of the course, this incredible mixing of microbes and humans and environment, it's going to make a lot of sense to you. All right, I will stop this video and we'll try to have a few more to finish the intro to the microlab. And again, I'm going to ask that you watch these videos at home so when we do our labs, you won't have to sit listening to me talk for hours and hours. We can just dive right into the fun stuff. Okay, folks, we'll see you next week.